Hello students, welcome to EPG Pathshala. I am Farhan Jalis Ahmed from Hamdard University, New Delhi. Today we are going to discuss on the module Colloidal and Supramolecular Drug Delivery Systems, which comes under the paper Novel Drug Delivery Systems Part 2. Our objective of this module is to understand the introduction of colloidal and supramolecular and drug delivery systems, to learn about various types of colloidal systems, to learn about various vesicular systems, and to understand their basic properties. Now, the particular or vesicular dosage forms, it is made up of polymers or lipids which are in the size range of nanometers to micrometers, and they are referred as colloidal drug carriers, CDCs. When these dispersed phase contains the particles in the size range of about 1 to 100 nanometer, the system can be called as colloidal system. These colloidal drug carriers can be transported across the various membranes, can be targeted to various sites containing drug, and it can improve their therapeutic index. It can also enhance or reduce their toxicity. The supramolecular systems is basically as a self-assembled systems in which the complex structure can be created when the molecules are bonded together by means of a non-covalent interaction. The CDCs, they can lead to improvement in the bioavailability, especially in the case of poorly water soluble drugs, and even it can enhance the absorption behavior. These carrier systems, they are also available in the market. For example, the liposomes which are available in the market in the name of ambisomes, which is a very popular drug containing an antifungal drug, and doxil, which is used for the treatment of Kaposi's sarcoma. Then there is a neural, which is widely used as an immunosuppressant, which is marketed in the formulation which is based on microemulsion drug delivery system. The CDCs and supramolecular system can easily protect the drug from degradation. It can enhance the drug absorption. It can modify their pharmacokinetic, and it can also improve the formulation. The CDCs or supramolecular systems can be administered by all routes of administration, and the bioavailability and therapeutic efficacy of the drug can be improved. The CDCs have been found very widespread applications in drug delivery, in gene therapy, and also in medical imaging. They have wide applications in various fields of pharmaceuticals, cosmetics, agricultural industry, biotechnology, and long-term conservation of fruits, vegetables, seeds, cereals, or fresh foodstuffs. Classification of colloidal carrier systems. These systems can be mainly classified as microparticulate systems, vesicular systems, supramolecular, and specialized emulsion systems. The detailed classification you can see in the figure. In addition to these systems, the resealed erythrocytes, antibodies, leukocytes, products, hydrogel-based nanoparticles, carbon nanotubes, mesoporous silica nanoparticles, solid-lipid nanoparticles also act as colloidal drug delivery systems. Most of the CDCs can be delivered to various sites, target sites in the body. Now, if you look at the classification, microparticles can be further classified as microspheres, nanoparticles, and nanoconstructs. And nanoconstructs is further classified as dendrimers and nanodiamonds. Talking about vesicular system, we have four types of vesicular systems: liposomes, neosomes, nanoliposomes, and pharmacosomes. While supramolecular systems, we have micelles. Micelles can be surfactant based, or micelles can be polymer based. Under the category of specialized emulsion systems, we have microemulsions and nanoemulsions. Both of them are same, only the main difference is their size and their composition. Now let's talk about microparticulate drug delivery systems. First, which comes under this class, is microspheres. Microspheres are small spherical shaped particles which are prepared using biodegradable polymers, and in which drug is filled homogeneously in the core, which act as a reservoir system for the drugs. Microsphere encapsulation protects the stability of drugs and make them more photostable. The drug can be loaded in the particles at two stages. Either it can be loaded during the preparation of the microspheres or after the preparation of microspheres. The microspheres can be utilized in gene therapy with DNA plasmids, release of proteins, vaccines, targeting tumor cells, and imaging. Now, if you look at the classification of nanoparticles, nanoparticles are small particles which can be classified as solid colloidal particles, polymeric nanoparticles, stealth nanoparticles, magnetic nanoparticles and metallic nanoparticles. These particles are in the size range of about 10 to 1000 nanometer. The drug can be loaded into these nanoparticles either by dissolution, by entrapment, or by encapsulation, or by attachment to a nanoparticles matrix, and may also be adsorbed onto the surface depending upon the properties of the surface. The drugs, proteins, DNA nanoparticles, they can easily permeate through the cell membranes and are stable in the bloodstream. Now, if we talk about polymeric nanoparticles, the nanoparticles can be made from natural or synthetic polymers, and they are called as polymeric nanoparticles. The most commonly used polymers are chitosan, albumin, and polylactide coglycosides, and are the most commonly used. Now, other class of nanoparticles is stealth nanoparticles. Stealth, the name stealth says they are invisible. The surface of these nanoparticles can be modified by means of coating the surface with hydrophilic polymers like polyethylene glycol, resulting in the formulation of stealth nanoparticles. It means they are invisible to our defensive mechanism, which is RES. These nanoparticles can have the ability to circulate for a prolonged period of time, and they can target the organ, because when they are circulating in the system for long period of time, their chances of bioavailability, their chances of accumulating at the organ is increased. Now, as a result of excellent drug loading, we can achieve long shelf life and control the release properties. These nanoparticles are found to be suitable for medical diagnostics, photothermal therapy, drug delivery, and nucleic acid delivery. Another class of nanoparticles is magnetic nanoparticles. The term magnetic means they are magnetically charged. These nanoparticles are made of using magnetic field gradients, and these particles consist of magnetic elements such as nickel and cobalt and their chemical compounds. When number of magnetic particles are joined together to form the clusters, these systems, they are called as magnetic nanobeads, which are having a size range of about 0.5 to 500 micrometer. Now, if we talk about metallic nanoparticles, the metallic nanoparticles known as gold nanoparticles in the size range of about 20 to 50 nanometer. Similarly, silver nanoparticles are in the size range of about 1 to 100 nanometer, and they show the most efficient cellular uptake. These metallic nanoparticles, both silver and gold nanoparticles, they have unique optical, physical and chemical properties, and which make them very suitable for enhancing the efficiency of drugs. The magnetic nanoparticles or supermagnetic iron oxide nanoparticles are best suitable for tumor treatment because of higher targeting efficiency. These nanoparticles have also been applied in the field of medical diagnostics and treatment of cancer as a catalyst in medical imaging, in genetic engineering, and as optical filters. Now, if you look at in the figure, the magnetic core, which consists of iron oxide at the center, which is magnified, and it is coated with a fluorescent dye and chitosan coating, and this is encapsulated in a lipid bilayer. The drug can be attached to the surface of the lipid bilayer, and because it is magnetic in nature, it can be attracted or it can be concentrated in area inside the body by applying an external magnetic field. So because of external magnetic field, all these particles, they move towards the tumor or towards the cell where we want to target, and there it will enter the cell and can release the drug carrier, and the drug will be released and the cell or tumor will be killed. So there we can target the magnetic core containing the drug into the cell, and other cells or other tissues or other parts of the body, they will be undisposed or unexposed to these drugs, because these drugs can be highly toxic in nature. These metallic nanoparticles, they are available for conjugation with proteins, DNA and amino acids. The widespread application of metallic nanoparticles also includes molecular imaging, diagnosis and delivery of nucleic acid and active pharmaceutical ingredients to target site. These nanoparticles are very commonly used for targeting these drugs, especially anti-cancer drugs, to various targets or various tumors inside the body. Now, dendrimers is a very novel class of structurally repeated branched macromolecules. that radiates from central core and are mainly derived from branches upon branches. They are tree-like structure, three-dimensional, regularly branched, proportioned core shell structure with a core, branches and functional end groups. They conjugate with various functional groups depending upon the nature of their branches. If we look at the structural arrangement of dendrimers, it has a very small core where you can have small molecules, nanoparticles or polymers. Then it has wide spaces between the branches. These wide spaces, they provide room for gas molecules. Gas molecule can get entrapped inside these wide spaces and will be protected from the external environment. Then you have surface groups which can be cationic, anionic, neutral, hydrophobic, biocompatible, targeted directing groups, biomarkers. So these surface groups, they can have various types of attachments. They can also define the properties of dendrimers, where they are going to attach, where they are going to target. Then if you look at the interior branches, they are robust, they are covalent structures, and they are connecting core to the surface. The dendrimers are classified according to their number of generations. After the core formation, addition of each stepwise monomers, next generation of dendrimers is generated. This method has ability to control the molecular weight and structural design of the dendrimer. And by controlling the molecular weight and structural design, we can control the amount of drug which can be encapsulated. We can control the target site of the dendrimer, and it can be used for various applications. Dendrimers can have both positive and negative charges, so that can make complexes with different types of drugs. Dendrimers are used in different fields like imaging, gene transfection, non-viral gene transfers, and drug delivery as a diagnostic tool for arteriosclerotic vasculature. Nanodiamonds are a very interesting type of nanosystems. They are carbon nanomaterials which are condensed octahedral diamond-like structure, which are in the size range of about 1 nanometer to 150 nanometer. And they are mainly used as a delivery vehicles for macromolecules, biotechnology products, and for small chemical drugs into cellular system, because they have a stability part which is very stable inner core. The particle size is also controlled. They are biocompatible. Their surface is alterable chemistry. They have reactive surface. There can be fluorescence, and they have the ability to form various types of hydrogels. Based on their particle size, nanodiamonds can be classified into diamonds where the particle size is in the range of 1 to 2 nanometer, or ultra nanocrystalline particles where the particle size ranges from 2 to 10 nanometer, and nanocrystalline particles where the size is between 1 to 150 nanometer. Detonation nanodiamonds have been potentially applied for the targeted delivery of different substances due to their high colloidal stability in aqueous and non-aqueous media. Nanodiamonds are mainly used in biomedical applications owing to its biocompatibility, commercial availability, stable photoluminescence, and minimal cytotoxicity. Nanodiamonds can enhance the absorption, durability, and therapeutic effectiveness. Nanodiamonds have also been applied for the treatment of gums, and therefore they are formulated in toothpaste and other cosmetic preparations like nail polish, eyeliners, shampoos, and facial lotions. If you look at the structural arrangement of nanodiamonds, they have functional layers, and it is coated outside with protective layers. An outside protective layer can be used for antigen detection, they can be used for fluorescent signaling, they can be used for shape recognition, they are biocompatible, and they also can be attached with linkers which can target it to a specific cell surface or a specific tissue surface. Liposomes are a very popular form of drug delivery system. When we talk about vesicular drug delivery systems, they are composed of one or more phospholipids layers which are arranged to form a vesicle, a spherical vesicle, in the size range of about 100 to 1000 nanometer. They are made up of cholesterol and phospholipids. These vesicles, they can encapsulate drugs, they can encapsulate vaccines within its walls, or layers wherein the aqueous compartments, and they may be encapsulated by a fatty envelope. The functional groups and pH of the medium play a key role in the formation of neutral, negative, or positive surface of the liposomes. and these can easily encapsulate both types of drugs, that is lipophilic and hydrophilic drugs. Liposomes can be used to deliver proteins, chemotherapeutic agents, RNA, DNA, and other therapeutics for the treatment of cancer, infections, and meningitis. Their advantages include low toxicity, reduced side effects, and enhanced efficacy of the encapsulated drugs and biocompatibility. The disadvantages are the rapid removal of conventional liposomes from the blood circulation due to recognition of surface-bounded opsonins by the mononuclear phagocyte system, and membrane license of charged liposomes by complement components, which impede the liposomal abilities to target to the other cells and organs and cause operation of delivered drugs into phagocytic cells. Neosomes are also spherical vesicles. They are composed of non-ionic surfactant, small spherical, and they are having a bilayered membrane which is composed of natural or synthetic amphiphilic lipids. They are mostly composed of non-ionic surfactant and cholesterol with or without the incorporation of lipids. Polyoxyethylene acetyl ethers, which is trade name known as bridge 58, and polyoxyethylene stearyl ethers, which is bridge 72 and 76. They are most commonly used in the preparation of neosomes. The hydrophilic end of the non-ionic surfactant, which is a major component which give it an advantage of being more thermodynamically stable outwards, and the hydrophobic ends face each other so that the bilayer formulation occurs in the neosomes and can easily deliver hydrophilic drugs in its aqueous core and lipophilic drugs in the bilayer made up of surfactants. Neosomes are discovered as alternative drug delivery system to defeat the problems which are mainly associated with sterilization, stability, and large-scale production of liposomes. They are biodegradable, biocompatible, and non-immunogenic in nature. The neosomes drug delivery systems have been exploited for their maximum therapeutic utilization in the management of drug targeting, gene delivery, anti-neoplastic treatment, transdermal drug delivery, and cosmetics. Nanoliposomes, the term nanoliposomes is used to describe the bilayer lipid vesicles which are very small in size range. In nano range, the particle size of nanoliposomes lies below 50 nanometer. They are mainly used for the encapsulation and delivery of nutrients, enzymes, food additives, and nutraceuticals. The lipid and the phospholipids are the main chemical ingredients of the nanoliposomes, which release their content in the presence of specific cellular enzymes due to the change in pH or change in temperature. Nanoliposomes can improve the bioavailability and stability of bioactive molecules and prevents the unwanted interaction with other molecules as well. The simultaneous addition of cholesterol and phospholipids in nanoliposomes results in enhancement of the stability by avoiding crystallization of the phospholipids. Pharmacosomes are the colloidal dispersions vesicles or in the form of hexagonal aggregates which are attached covalently to phospholipids and having a particle size range of about 200 to 250 nanometer. These are neutral molecules having both positive and negative charge. They are water loving and fat loving both properties. These drugs are conjugated by electrostatic forces or by hydrogen bond by formation with lipids. The pharmacosomes have the capacity to augment the drug absorption and its transport across the biological membranes. These can improve the rate of permeation of drug by improving membrane fluidity. They can target tissues and have wider stability profile. The pharmacosomes have been widely used for the delivery of drugs, used for cancer, heart diseases, inflammation, and delivery of proteins and herbal drugs. Let's talk about micelles. Micelles are spherical structures, you can look in the figure, with amphiphilic nature poised of a non-aqueous core and aqueous shell. Micelles of amphiphilic copolymers are used to encapsulate hydrophobic organic molecules and polymers to improve their effects in biological environment. Polymeric micelles are nanoscopic core shell structure with a size range of 10 to 100 nanometer, and they are formed by amphiphilic block copolymers. You can see in the figure, both are explained here, polymeric micelles containing amphiphilic block copolymers and lipophilic substances. Similarly, hydrophilic polar head, phospholipid molecule, and hydrophobic non-polar fatty acid tail. Several water-soluble polymers, polyethylene glycols, poly-N2-hydroxypropyl, methacrylamides, poly-L-glutamic acid, and dextran have been utilized successfully in the formulation of missiles. They are used to deliver a large number of products because of their size, high monodispacity, surface functionality, and stability. The drug is packed into amphiphilic micelle structure by two methods, that are drug conjugation and drug encapsulation. In drug conjugation, the lipophilic drug is used as a hydrophobic core which is conjugated with the hydrophilic polymer by biodegradable chemical linkers for effective release of the drug. In drug encapsulation, the drugs are actually confined into the hydrophobic core of the micelles. Stop. Micelles are biocompatible, and due to their nano size less than 50 nanometer, they remain in the blood circulation for a long period of time, and their hydrophilic shell resists their eradication by the reticuloendothelial system. Supraparamagnetic iron oxide nanoparticles or polymeric micelles, they are used as a MRI probes and for drug delivery. The drugs can be targeted to poorly permeable tumors and cancers by making the micelles stimuli responsive. Micelles have the capacity to increase the solubility of hydrophobic drugs, and hydrophilic shell avoids the recognition and opsonization by RES. The polymeric micelles are used for the delivery of anti-cancer drugs and also as a diagnostic imaging technique in the form of nanomedicine, used to deliver the polymers into cells for bioimaging by encapsulating the hydrophobic conjugated polymers into their core shell structure, used as suitable ocular carriers for anti-inflammatory drugs, plasmids, and genes. Microemulsions, which are coming under specialized emulsion systems, they are clear or translucent systems which are having a droplet size of about 150 nanometer. These compose of a blend of oil, water, and surfactants, and are thermodynamically stable and optically isotropic. The interfacial film of the surfactant and the co-surfactant molecules result in the stabilization of microemulsion. These can be used as a potential drug delivery system, either as a vehicle or topical application, or for the enhancement of viability of poorly water-soluble drugs. Microemulsion are being utilized in the field of agrochemicals, cosmetics, biotechnology, and as well as in drug delivery. Nanoemulsions. Nanoemulsions consist of an aqueous phase and emulsified oil with mean diameter in the range of 50 to 1000 nanometer. If you look at the structure of nanoemulsion, there are oil droplets at the center which are surrounded by emulsifier. In emulsifier, hydrophilic head is facing outside and hydrophobic tail is facing inside. So your hydrophobic drugs which are soluble in oil, they can be encapsulated inside the core, and because of the hydrophilic head, it can be easily dispersed into an aqueous phase. Nanoemulsion is an appropriate technology to encapsulate various compounds like drugs, antimicrobial agents, nutraceuticals, antioxidants, and flavors to prevent their degradation and improve their bioavailability. They are the dispersions of two non-miscible liquids having dispersion of oil in aqueous phase, oil in water nanoemulsion, or dispersion of aqueous phase in oil, water and oil emulsion. Nanoemulsion can help in the delivery of both hydrophobic and hydrophilic drugs by converting them in both water and oil or oil in water nanoemulsions. The surfactants which are used by the industry, they are mostly grass category, and nanoemulsions have widespread applications in the field of vaccine delivery, in cancer therapy, and target delivery and ocular delivery. These have various applications in cosmetic delivery of gene and phytopharmaceuticals and improved delivery of poorly soluble drugs. So students, let's conclude what we have learned from this module. We have learned about colloidal drug carriers. As these carriers, they are widely accepted as a drug delivery systems, and very few of them are currently available in the market. These systems are widely used due to their controlled release of active medicaments. They can prevent the degradation of drugs. They have very high interpret efficiency. They improve the therapeutic efficacy of APIs and excellent drug targeting. We can target these drugs to various organs, to various sites. We can increase their residence time inside the body, especially after the introduction of stealth liposomes. We have understood that stealth liposomes and nanoparticles, they show diminished phagocytosis. Because of diminished phagocytosis, they are circulating in the body for a very prolonged period of time. And when they are circulating in the system, in your blood for prolonged period of time, they can be absorbed by various organs, various tissues, depending upon their surface properties, and their targeting will be more efficient. So their targeting is improved. Now in future, these colloidal carriers, they will be useful in the formulation of biotechnology based drug like proteins, genes, or oligonucleotides. These oligonucleotides, because nowadays most of the systems which are coming into the market, they are biotechnology based. We have less of chemical based drugs and more of biotechnology based drugs, and these drugs cannot be given by oral route, because when they are taken orally, they are being degraded. So CDCs are the, I think, is the future of these biotechnology based drugs, and we can use them for various applications.